Yeah, without further ado, I think we'll just, Zaza can kick off the talk. I'm sure most of you know Zaza. Zaza is a lecturer in the department. He has his research interests are in spatial machine learning, ecological statistics, and species distribution models. I hope that's sufficient. Zaza, you can.
Thank you, Craig. I don't worry, I know technology can be a pain sometimes. Okay, so my talk is on constraint ordination. It's a light talk. It's light on the theory, both ecological theory and statistical theory. So I've tried to focus mostly on the R functions, so the R functionality. Okay, it's very short talk. A toolbox on multivariate analysis methods. Craig, can you please record? We've had a toolbox on multivariate analysis, so I can just point you out to that toolbox. So it's a nice introduction of MVA in ecology, and it's very broad. Okay. So cluster analysis seeks to find discontinuities in dataset, such that items in a cluster are similar and items between clusters are as different as possible. So in cluster analysis, we basically want to find groups of similar items, which is commonly sites, and we want them to be grouped together such that sites which are in one cluster are very similar, and sites which are in different clusters are markedly different. The other technique for multivariate analysis in ecology is our ordination. Ordination, on the other hand, it doesn't look for discontinuities, but it actually seeks to extract the trends in the data set in the form of a continuous axis. So the whole aim of this is to represent the relationships in your multivariate data as faithfully as possible, using fewer dimensions than there were in your data sets. So you might have, say, take, for example, our R matrix, where you have sites and different environmental variables. One way of viewing that data set would be to try and plot the sites in that highly dimensional space with all your environmental variables. But then it might be hard to visualize the patterns in the data set. So what we are seeking to do with ordination is basically to reduce the dimensions of our data set so we can better inspect the patterns in our data set. So we usually try to reduce our dimensions to 2 dimensions and sometimes 3, but hardly. So in reducing your data set from your high dimensional space to your lower dimensions, you would end up with a 2D plot. You could either have on this 2D plot, basically, you would have to locate your sites on that plot. So here it will tell you which that sites close together are similar in terms of species compositions. Okay? On that same plot, you might also add not only the sites, but you might also add the species. So then you can even tell for which sites are more associated with a particular species. So these sites over here are associated with those species over there. So these are hypothetical examples, so there are no names. Another plot, which is still an ordination plot, is where you would have your sites, your samples. So you'd have your samples there, the different species, and you'd have some variable plotted in that two-dimensional space. So eventually, when you're doing an ordination, this is what you're looking for, a visual plot, which you can then inspect for patterns in your data set. Yeah. So, back. From a practical point of view, we seek to extract and visualize environmental gradients from our ecological data, which are then used as a basis for describing the differential habitat preferences. So we want to know what kind of sites basically our species prefer, and which species prefer similar sites, and which sites are similar. So with that plot, as you can see from such plots, you can get that information in terms of which sites are similar, which species are associated with which particular sites. So from a practical point of view, with our ordination, we seek to either describe the community pattern, or we want to explain and test changes in species composition via some variables, usually our environmental variables. So the first task of describing the community pattern can be addressed via unconstrained ordination, also known as indirect gradient descent. So here, unconstrained ordination will basically locate your points, which are your samples or which are your species, in your reduced dimensional space, okay? But it doesn't try to, it doesn't give explanations, and you cannot say anything in terms of significance in terms of, with regards of those patterns. So that is an unconstrained ordination. So we're looking at the second method, which is constrained ordination, also known as direct gradient descent. So constrained ordination allows us to actually test what we're seeing from our ordination plots, and actually test each of environmental variables to see which of them have the greatest effect when it comes to the patterns we are observing. So it's just one more higher level of analysis you can do with constrained ordination versus our unconstrained counterpart. Okay. So we've said in multivariate analysis of ecological data, there are two branches there: your cluster analysis, hierarchical or non-hierarchical, or your ordination methods. And we've already identified that in ordination methods, they are unconstrained methods and constrained methods. So in our unconstrained methods, we've got a single data set. We perform some ordination on that data set, and we try to interpret the patterns we're seeing there a posterior. So after we've done the analysis, okay? So methods which fall under unconstrained ordination are your principal component analysis, correspondence analysis, and multi-dimensional scaling MDS. Then we said in our constrained ordination, we are trying to relate to data sets. So we're trying to relate our data of species compositions, that is our L matrix, to a matrix of environmental variables at the different sites. That's our R matrix. So we've got two data sets there. You can have more, but the starting point is always two. So examples of constrained ordination methods are RDA, which is redundancy analysis, and CCA, which is canonical correspondence analysis. Okay, so we've seen what constrained ordination is. What is it doing practically? So constraining an ordination means that the ordination only shows community variation and can be attributed to some descriptor variables. So we're working in an environmental space here. So our descriptor variables are usually environmental variables. So obviously this means that in the unconstrained setting, we are just trying to find directions of maximum variation, but those directions do not necessarily correspond to any environmental variable. So we can think of them as being synthetic environmental gradients. Okay. So let's just conclude there that in unconstrained ordination, we've got one data matrix, and the method is descriptive, and we have no statistical tests to assess the significance of any patterns that are detected. Whilst in the constrained setting, we're able to explore relationships between multiple data matrices. So as an example, our L matrix and our R matrix. And also with constrained ordination, we're able to test the significance of relationships we are observing. Okay? So we're able to test the significance of relationships between our environmental variables and our species compositions. So that's just the very general setting of ordination. Let's look at a quick, not so detailed look at constraint ordination. So we already know what it is. We're constraining our ordination with regards to some environmental variables. What is actually happening at the back? So constraint ordination can be thought of as a set of multivariate regression analysis in which each set of variables is regressed against each sample. Okay, but I'll show you a graphic there, which might communicate that a bit better. We note that because of the link to regression, constraint ordination allows one to decompose the total variance in species composition data, that's our L matrix, into a fraction explained by environmental variables and a fraction not explained by environmental variables. So the fraction explained by environmental variables is basically our constrained ordination axis, and that one not explained by environmental variables. That would be our unconstrained ordinational axis. So I think you can already appreciate that in constrained ordination, you are basically performing both unconstrained and constrained ordination in the same method. Okay. So constrained ordination allows us to relate the species composition directly to our environmental variables and extract the variance in our species composition data, which is directly related to the environment. So instead of just extracting directions of maximum variation, we're extracting those directions in relation to environmental variables. So hence our constraint ordination axis correspond to the directions of variability in the data, which is explained by environmental variables. That's what I've just said. Okay. Two of the most widely used methods are redundancy analysis and canonical correspondence analysis. Good time. So let's just have a quick overview of what is happening with redundancy analysis. We said ordination basically employs some multiple linear regression in there, which is what is allowing us to decompose variation to one variation explained by our environmental variables and variation which we cannot explain. So what is happening in an RDA algorithm? You've got your data set there, sample species matrix there, which is our L matrix. And we also have our R matrix there. It's only got one variable in there, as an example. And we want to relate this matrix of sites and species to our matrix of environmental variables. So the first thing that is done is that we regress, that's for each of the species, we regress those values against our environmental variable. So we try to predict the amount, if we have abundances in there, So we try to predict the abundance at each sample site for each species, okay? And that is our first regression. So then obviously we'll have a matrix of predicted values. So this will be the predicted abundances for each of the sites for that particular species. So this is repeated for all species in your data set. So that's what has happened here. So we've done the regression on each of the species, all of the sites, and we end up with a matrix of predicted values. Okay. Where we've got a regression, we obviously have some residuals. We know residuals are our predicted values, the difference between our predicted values and the observed values. So from the regression, we also get out a matrix of residuals for each of the sites and species there. Then the RDA algorithm finally then takes that matrix of predicted values and performs a PCA on it to give us our constrained ordination axis. And it also performs a PCA on the residuals to give us our unconstrained ordination axis. So it's fairly straightforward. You can see what is happening. Okay. Craig, I see something saying the meeting will end in 10 minutes.
Yeah, that's odd. Shouldn't we have?
I've got a whole license, I don't know.
Yeah. Okay, we'll just come back to this link if and when it ends.
Okay. So that would be our RDA algorithm. So we just have an idea of what's happening at the back without looking at the mathematics. Yeah. So CCA, or canonical correspondence analysis, works in a similar way to RDA, but here it combines multiple regression with correspondence analysis. Again, if you're unfamiliar with correspondence analysis, I would refer you to our previous toolbox on multivariate analysis. So just have a look at that. But we note that the algorithm of RDA is modified such that instead of our raw species composition data, instead of that raw L matrix, the set of regressions is done on the Q matrix. So as our Q matrix is our species traits matrix, and the weighted multiple regression is used instead of simple multiple linear regression. Okay, so we're weighting using the row sums. Okay. It's a nice detail, but it's not so important for us. Lastly, we note these two methods, RDA and CAA, basically are best used for different settings. Okay, so RDA is best used for exploring linear responses to environmental gradients, whilst CCA is best for investigating unimodal relationships. So I've just put a graphic there at the bottom to explain what's a linear and a unimodal relationship. So in a linear environmental gradient is basically where you'd expect a linear relationship between your variables and your abundances, okay? But what is common in nature is that there's some optimum value for an environmental variable, say temperature, at which your species may thrive, so your plants or your birds may thrive. Then at lower or higher values of that temperature, your species doesn't do so well. So this is a unimodal relationship, and that is actually what we'd expect to see in practice. So you'd have to look at your data set and decide if it's a linear or unimodal relationship. So I have not covered it here, but there's a rule of thumb and a test for deciding if you should use either linear ordination methods or unimodal methods. Okay, so that's all the theory. Maybe we should look at an example to try and see what you can actually do with constraint ordination. So we're going to use data from the R package vegan. So if you're interested on knowing further about the data set here, I've given the paper in which it's first used. So effects of reindeer grazing on understory vegetation there. So you can have a look at that paper if you want to know more about that data set. But in summary, the data set, or that reindeer data set, consists of two parts: an L matrix, so our species, our samples, species composition matrix, where we have sampled at 24 sites, and we're trying to observe what's happening with those 24 sites for 44 species. Okay, so each of the sites, for each of the species, we've gone and looked how much cover, what's the percentage cover for each of the species. Okay, so you take a site and say it's 50% covered by a specific species, 30% covered by a specific species. And that's what goes into our L matrix. Our R matrix, so matrix of environmental variables, contains 14 continuous environmental variables observed at our sample sites. Okay, so your R matrix doesn't necessarily have to be strictly made up of continuous environmental variables. You can also have categorical variables. So maybe land classes or something of a sort. So the method, so constraint ordination accepts both continuous environmental variables and categorical or factors. But here our example only has continuous variables. Okay, so preliminaries. You would have to load your vegan package, then we get our data. So let's first look at our L matrix or sample by species matrix. You can see that I've just printed the header of the first few values or first few rows there. So you can see that the columns are giving you the different species down there, and the rows are the different sites. So that's site 18. Then for that species, we've observed 0.55% cover. Okay, so some days a 17% cover for that. So that's for each of the sites. So site 24, also those are the percentage cover values for our different species. Okay, so this matrix is how it should be for an L matrix. Let's look at our R matrix. So our matrix of sample sites by environmental variables. So we know we've got 24 sample sites and we have 14 variables. So if you have look at the structure of the variables, you already know that they're continuous. So you can see that they're all numerical variables, and those are the names of our variables. So most of them are chemical properties, so minerals. Bare soil is just the percentage bare soil. Humus depth, which is basically the depth of the humus at each site. Then pH just relates to how alkaline or acidic the soil is at that site. Okay, so let's try and analyze this data set. Let's start with an unconstrained ordination. So unconstrained ordination, so we're just taking our species composition matrix and trying to see which of our sites are similar to each other, okay? So we're not constraining this ordination. If you use the function RDA and you just give it one data matrix, then it performs unconstrained ordination, okay? As it says, the eigenvalues for unconstrained axis. So this is the unconstrained ordination, and as your output, you'd get information on the inertia. So inertia relates to the variance for a multivariate data set. Okay. Then you also get out your eigenvalues for each of our axes. So these eigenvalues are arranged in descending order, with the first component explaining the most variance and others explaining subsequently less. Okay? So the eigenvalues relate directly to the amount of variation explained by that axis. And we can see how much variation is explained by axis 1 and by axis 2 and the subsequent ones. So remember, our total variation is 1826. So if you take all the dimensions of your principal component axis, you should be able to explain the total variation in your data set. But usually you do not want all the components, you just want a subset of them, and you want them to explain as much as possible of that total variation. So let's look at the plot we obtained for our unconstrained ordination. We can see but I think it cut at the proper place. We can continue from here. Okay, so what we're trying to do now, so we've performed an unconstrained ordination. Let's see what our plot gives us. So we remember the plot is supposed to reveal patterns in terms of our sites. Then we can see these numbers here, the different site identifiers, and we can see which sites are similar and which are different. Sites 9 and 10 are quite similar, and they seem to have high values of that species, the pedestal, okay? And so are sites 12 and 2, they've got high values there. And then sites 5 and 7, 6 are also similar sites in terms of species composition and of high values for clad rank. Yeah, so from our unconstrained ordination plot, we're able to get some information from that to know which of our sites are similar, but we cannot really test the patterns we are observing. We can only just describe what we're seeing. Okay, so that was the unconstrained ordination setting. So we want to constrain our ordination now, and we're still doing redundancy analysis. So the analysis I'm doing here with redundancy analysis is very similar with what you would do with canonical correspondence analysis. So the function for CCA is CCA. So where I have RPA there, you'd have CCA. But the functions basically do the same thing, perform the same task and return the same output. Okay, so we're trying here to perform a constrained ordination. So what are we giving our RDA function? We give it our data matrix of species compositions, so our L matrix, and then we also give it the matrix of environmental variables. Okay. So here I've used a formula expression saying that I want all the variables in that data set to be included. Okay. You could also type in the individual variables you're interested in. So if you just want potassium, you put it in there. If you want calcium, so you just type them in there. But here, as a starting point, we're using all the environmental variables. Okay. So it's best to do it this way via a formula, but RDA also just takes, you can just give it the L and your R matrix in this form over there, and will still perform your RDA. But it will not take, if you perform your RDA in this form, some of the functionality for the analysis is not available. So it's always best to do it via the formula expression. So we've performed an RDA. We've constrained our ordination using our 14 environmental variables. Let's see what we get out. As before, we get out some information. It's telling us about the total inertia in our data set, how much of the inertia, that's the variation, is explained by our constrained ordination, and how much is explained by our unconstrained ordination. Okay? We also get eigenvalues for our constrained axis. So we know what eigenvalues are as before. Eigenvalues relate to the amount of variance explained by each of those axes. So we have eigenvalues for our constraint axis and also eigenvalues for our unconstrained axis. Remember, RDA performs both of these constraint and unconstrained analysis. Constraint ordination as it executes the algorithm. Hence, we have output for both an unconstrained and a constrained ordination. Okay, so if you want, you might call summary to your fitted RDA objects, then you get much more information out. So most of the information is not really useful, but it's used by R to do the plotting for you. Unless you're a statistician or interested in other methods, you may not even look at that output there. So what you're getting is eigenvalues telling you importance of components, the accumulated eigenvalues for our components, species scores. So we use these species scores to plot the species on our reduced dimensional plot. So if we're using 2 dimensions, we'd use the species scores from the first 2 dimensions to locate each of the species. Also the sites, remember we can also plot sites in our ordination plot. So if we're working with 2 dimensions, we'll use these first 2 axes to plot the sites. Okay. So there's a lot of information that you can get out. But let's look at our plots, which is the main aim when you're doing an ordination analysis. So now we've done an ordination analysis. Here we've got a little bit more constraint ordination analysis. Here we've got a little bit more information than what we had for the unconstrained ordination. But first, the first thing you should note is that our axes, so remember axis 1, axis 2, they correspond more closely to our environmental variables. So you can see our first axis correspond to bare soil. So that's what we want. We try to force the ordination such that we get the variation as restricted by our environmental variables. We do not just want variation, we want that bit which is explained by our environmental variables. So bare soil seems to explain the most variation there, as it is associated with our first axis. And our second axis seems to be associated with P and S and sulfur there, but not so much. Your first axis will always associate more with one of your variables. So we've got more information in there. So we've got our environmental variables in there. We've got our sites, and we also have the different species. So you can have similar information there to sites 5, 7, 6 are similar the high on clad rank species, and these sites also have high values for nitrogen over there. Okay, so the direction of your variables basically is telling you, or maybe I should put it this way. The sites, you want to look at each of the sites and see which variables are pointing that way. So you can tell like site 3 is highly influenced by pH and iron over there, and Al, aluminium. Okay, then sites on this side would have lower values for these variables. And a similar 18 and 13 have high nitrogen values, and the sites here will have lower nitrogen values. So we're able to get quite a lot of information out from using a constraint ordination. But it's okay, we haven't done anything yet. So we just basically have explored. We haven't performed any significance tests, and we know that's the strength of constraint ordination. So firstly, we want to see if our model is any good at all. Okay, so we calculate the adjusted R for our model, and we find it to be 0.487, with the R squared being a higher value. So remember that constraint ordination uses multiple linear regression within it. And we know the problems of multiple linear regression, that if you add more variables, you always will explain more variation, but that's not what you always want. So a more honest, I would say, statistic is the adjusted R squared, which controls for the number of variables in your regression. Okay. So then if you're using adjusted R squared, then you are able to compare regressions with different amounts of or numbers of variables. So when you're reading or you're interpreting the significance or ask the significance of your whole model, always use your adjusted R. Yeah. Okay, we get a value of 0.488 there. We want to know that significance seems low, but is it statistically significant? Okay, firstly, we shouldn't be surprised that we get such a low significance. Remember, this is multivariate data, it's highly dimensional data. So being able to explain around 48% of the variation is quite good already. So what we want to see now is that model significant or not. So the significance test is performed via the function ANOVA.cca. So this ANOVA.cca will perform significance tests for redundancy analysis and also for canonical correspondence analysis. Okay. So we perform our significance test and we find that our model is indeed significant, which means we can go ahead and use our model. So, but that's not the end of it. Remember, in the end, when we are reporting our results, we'll use a two-dimensional visualization. So we basically want our first two axes to be significant. Okay? And if we look at the significance of the axis, so we're able to test the significance of the axis by specifying that argument for your ANOVA.cca call there. So we're saying we want to test the significance of the individual axes. And when we do that, we find that only the first axis is significant and the other axes are not significant. What we're really interested in is axis 1 and axis 2 being significant. Okay? And we see that we've fallen short here and our second axis is not significant. Okay. We may also be interested in exploring the significance of our terms, so our individual environmental variables, if they are significant or not. First test is a significance test where you consider the sequence in which you've entered the terms. So your N comes in first, your P comes in first, and you can perform a significance test there. But this one is not ideal because it depends on the order in which your terms enter your expression. So a better significance test for your variables, your explanatory variables, is one which does not depend on the order of the terms. And if you specify by margin when you're calling ANOVA CCA, that is what will be performed, a significance test where we have ignored the order in which your terms enter the model. Having done that, we find that humus debt is significant, MO is significant, forget what symbol, what element is that? Sulfur is significant, and potassium is significant there. So those are our significant environmental variables over there. Okay, so we saw that our fit was significant, but our second axis was not significant. So we'd like to improve upon that so that our final ordination plot consists of both axes being significant. So maybe we must remove some of our variables in there. So that's what we're trying to do. We are performing a variable selection. First, we specify a non-model with just the intercepts there. So there's none of our environmental variables in there. And then once you've specified that non-model, you can use the function ODR2 step then to perform stepwise selection of your variables. So the selection might be either forward or backward or both. So remember, this is related to multiple regression. So in regression, you might take a variable, put it into the regression and see if it's an improvement over the model without that variable. So that's essentially what's happening here. So you can specify the direction of your variable selection. So you're starting with all variables and going backward, removing some, or you're starting from your non-model or adding them inside there. So that is your forward selection. So I prefer both forward and back selection. Okay. So another thing you should note about that ODR2 step is that you also have to specify what you are willing to accept as a good model. So we've specified that we want our model to be at least as good as our model where we had everything in there. So that adjusted R with everything in there. So once then you let the function run, and you see that the function settles on 3 variables, or 3 environmental variables as being the best variables for predicting our species composition: aluminum, potassium, nitrogen, and the soil over there. Okay. And we get this same information as we did before, the inertia, our eigenvalues for the different axes, our constraint and our constraint axis. Okay, so it seems like the model with the aluminum, potassium, nitrogen, and best soil is the best model. So let's test the individual terms in our model to see if they're actually significant. We want them to be. So if you call ANOVA to your selection, your stepwise selection, it will tell you if your variables are significant, and it will also give you an adjusted R value for when each of those variables enter the regression. Okay. So we would be happy with this because we see that all our terms are significant. We also require that our 2 axes, our first and second axis, be significant. So let's check now if that is the case. So we perform our test for the axis, and you will see that the first and now even the second axis are significant, which is good, which means now our ordination plot is more meaningful. Since we know that both our first and our second axis are significant, and we know the variables which the significant variables are working in, then we perform the ordination using that. And this gives you the sites now in relationship to our 4 environmental variables, which were deemed to be the best discriminators. So that's what you have over there. Can we move the next slide? So this is our last bit. So you might also be interested in partitioning the variation you're observing to groups of terms. Okay, so the variation, we're talking about the total sum of squares here. We want to partition the total sum of squares to groups of terms. So we could think of aluminium, potassium, and nitrogen as being one group of variables, which are what our chemical properties of the soil, and we group them together. Then our other significant variable, the soil being some other components or characteristics of the soil. So we consider this as being related and that as being on its own. So we can see, we want to see how much variation can be attributed to the set of chemical variables and to that individual variable. So there we've partitioned the variance, and you can see, you can see how much variation is explained by each. So X1, which is our aluminium, potassium, nitrogen, explains 42% of the variation, with bare soil on its own explaining 18%. We shouldn't be surprised at the 42% there. We've got 3 variables explaining that. So you might also look at other fractions or quantities. And then you can also plot this in terms of a Venn diagram, which is basically reporting what you have over there. So remember, our X1 is comprised of our 3 mineral variables. And you see that if you add that and that, it will give you your adjusted R squared for that set of variables. And if you add this and that, it will give you your R squared for your bare soil. Okay. So what is this Venn diagram telling us? It's telling us that 0.26 of the variation can be strictly attributed to our 3 chemical variables, Al, K, and N, and 0.07 can be attributed strictly to bare soil. Then there's some variation, 0.08, which we cannot really disentangle and attribute to each of the groups of variables. So that's basically arising due to the correlation between our X1 variables and our X2 over there. So we can then not disentangle. So this is the shared variation explained by that 2 groups of variables. Okay, so there's a explanation there of what the different proportions are there. So as I was talking earlier on the B, so B is the shared variation explained by both variables that cannot be decided to which term it should be attributed to, and is a result of the correlation between our variables or sets of variables. Okay. Then you can also read out what is the different areas of our Venn diagram. So A, in passing, it's the variation explained by our variable 1. We know it's a group of variables in our case. conditional on the effects of our other variable. Okay, this is the amount of variation that would be able to be explained if we are considering variable 2 as a covariable, and C is something similar to that. And we go to our second to the last slide. We may also be able to, we may be interested in partitioning the variation to individual terms, but this is not of much interest for practical situations, because you can get the amount of variation from the individual terms with other outputs. But here I've just put it for the sake of completeness. And similarly, then you can get a Venn diagram which explains to you what proportions of the variation are explained by each variable. So our variable will expand on its own, is able to explain 15%, but there's shared variation also with the x3 of 0.1, shared variation also there, and shared variation over there. Our x4 variable is able to explain 0.07 alone, and those are the shared variations over there. Okay, so I hope that was informative. I've tried to limit the theory. Yeah, but numerical ecology, you know, it's a bit, it's one of the more math-heavy ecology courses over there. You can have a look at the book Numerical Ecology with R, where it shows you some of the code, shows you the code you'd use for such an analysis. Then if you are interested in the community ecology theory, there's Numerical Ecology, the book by Lagrange and his brother there. Also, yeah, I've used the package vegan to do all analysis. So that's just a reference for the package. Okay. 10 minutes.
Right. Thanks, Zaza. We'll now open the floor for questions. And I see we have a question from Shakira in the chat. She says, how would the ordination work when you have categorical environmental variables, e.g. substrate type? Would the impact be the same?
Yes, it would be the same. So what would happen? Let's look at where we're putting them in. Well, first we'll look here. Okay, let me say, okay, maybe we've actually stated what they are. Okay, there's no way I'm stating exactly what they are, but basically you would put them in here. So the continuous variable would just go in as is. Then if it's a categorical variable, you'd say factor, then you'd put in that variable inside that regression. So only thing you just have to wrap that variable with factor so that R knows it's a factor, it's a categorical variable. Then you just proceed as before.
Cool. And then we have Greg.
Thanks, Zaz. Just a simple question. You said a couple of times that when you do the constrained ordination, you kind of also get the unconstrained ordination at the same time.
Yeah.
But I just wanted to double check. Clearly, it will be different. The results you get from that unconstrained ordination would be different to a complete unconstrained ordination because it's an unconstrained ordination on the variance left over after accounting for the predictor variables, right?
Okay, well put, Greg, because you're working, you're basically ordinating the residuals. So okay, that's the amount of leftover variation.
Okay, cool. Thank you.
Cool. Do we have any more questions for Zaza?
Hi, Pachi.
Yeah.
Hi, Tata. Thank you. Thank you very much. So you've said multiple times that you have like a linear regression running in different steps. So I understand there would be problems with collinearity of variables and stuff like that.
Thank you.
Yeah. And is there any particular step where you have to be extra cautious?
Yes, I should have mentioned things I didn't touch on were issues of transformations. So maybe first of all, if your sample species matrix has some very rare species, you want to perform some kind of transformation in there before you perform the ordination. I think Watling is one of the more common ones. So I didn't touch on that. I also didn't touch on the issue of collinearity. So there is a function to perform collinearity test on your fitted regression or fitted ordination model. So what as we've done here, you'd fit all your data set. Then if you call VIF, so it's VIF.CAA, it's basically performing a variance inflation test on that ordination. Then you'd have to remove all variables with which are highly collinear. So usually cutoff is 10. So all values with the VIF values above 10 you would exclude from your regression, from your variable selection procedures over there. So yes, Pachi, it's a problem, and yes, you do need to consider it.
All right, cool, perfect, thanks.
Thanks. We have about 10 more minutes to go, so we can, we can sneak in one or two more questions. Do we have any takers? If we don't, yeah, the attendance for me was a bit disappointing. I hope it's load shedding and not Zoom fatigue or anything else. But when you do these invites, please do not hesitate to pass them on to people who may not be on our list. And if there aren't any more questions, I'm gonna stop the recording, and thank you very much for joining us.
Thank you, Craig. Maybe just to add, I think this is a very specialized topic, so most people maybe felt like they wouldn't be able to use it because it's a community ecology.
Yeah, that could be the reason why as well. But thank you very much, and apologies again for the hiccup in the beginning.
No problem, Craig. Thank you as always.
Thank you. Bye, everyone.
Cheers.